Good morning and welcome to Global Compliance Panel's live webinar on Auditing Analytical Laboratories for FDA Compliance. My name is Michael and I will be your host today. On behalf of the Global Compliance Panel team, I'd like to thank you all for being part of this event. And today's webinar is being presented by Dr. Stephen S. Guara. Mr. Guara, PhD, is the founder and principal of GXP Biotechnology, LLC, which is a consulting firm, and they work in the areas covered by the GLP and GMP of drugs, biologics, and nutraceuticals. Steve has over 30 years of experience in supervising quality control laboratories, including an animal testing facility, and in performing GLP and GMP audits of internal and external testing laboratories. Steve has participated in the development of drugs and biologicals through all phases of clinical research and the final product production as well. And we are honored to have Mr. Guara with us to present the webinar. And before we begin, I just want to quickly outline today's session information. This webinar is for a 90-minute duration. First, Steve will take you through today's webinar and highlight the areas that he would cover, and then he would share you his presentation. And also want to inform all of you participants that once part of this teleconference, you've been placed on mute and would remain so until the Q&A window begins, which is again at the end of the session. This is just to ensure that there is no discontinuity during the session and the presenter could speak clearly and all of you could take maximum benefit from the session. And also, you would be allotted about 10 minutes of time at the end of the session for any kind of question and answers. And also, if you do get out of the session, please follow the same procedure to log in again. And now that we're all ready to start, I request you to take it from here. Steve?
Thank you very much, Michael. Good morning or good afternoon, depending upon where you're located. We will go through the auditing of analytical laboratories, and I will warn you that we are primarily concerned with FDA compliance. Now, there are, of course, other agencies that may get involved in, well, depending upon where you're selling your products, you may end up looking at other jurisdictions. And what I am going to cover should be general enough that it will cover all of these other jurisdictions as well. Although I will warn you that each little jurisdiction does have its own peculiarities. So you do need to go over their regulations. Now, one of the problems in auditing a laboratory is that some people think of a laboratory as a service organization rather than a manufacturing organization. And so they think that they need to use different procedures and techniques. Actually, this is not true. You can look at a laboratory as a small manufacturing facility. And if you look at the functions of a laboratory and what goes on there, and we're primarily talking about testing laboratories, the whole process is very much like that of a manufacturing facility. And so you can easily find correlations between what you would do for a manufacturing facility and what you need to do when you look at a laboratory. Now, there are several different types of laboratories, of course, and each one of these will require a slightly different approach. And actually, I will kind of leave it to you to adjust your approach. Basically, you can look at independent laboratories as an example of the whole set. And the main differences would be whether you're looking at a straight physical laboratory or if you're dealing with a laboratory that does biological testing as well. Because with biological testing, especially if they're using whole animals, you will need to invoke the GLP regulations as well as the GMP. Now, if you look at the different types of laboratories, I will point out to you that there are government laboratories, and usually the government laboratories are not audited. However, a government may invite you to audit them, especially if they're testing your product, etc. Um, remember, there are no perfect laboratories. If you talk to them, they all tell you that they are, but you know, if you really found the perfect laboratory, then you're probably in another world. Now, some companies are going to protest. They're going to tell you, oh, we have certified analysts, or we have certified chemists and all this. Remember this, certified means minimum standard. The worker has met some sort of minimum standard. But nobody is an expert at everything, unless they're crazy. But the fact of the matter is that you want to make sure that they really are expert at doing what they're doing for you. And the other thing is that, you know, laboratories have customers and they have to respond to their customers. So, you know, and you're the customer, presumably, if you're auditing them. And remember, too, that a lot of times you will need to audit them before you sign a contract with them. There are some laboratories that you really do want to look into, because after the audit, you may decide you don't want a contract with them. Now, in setting up for the audit, there are certain niceties. They're done by all auditors, well, all trained auditors anyway. And these things should be followed for, you know, just about any audit. Now, getting into a hostile or an adversarial audit really, you know, is not valuable, because you're fighting resistance all the way. There's no cooperation. And the only time you want to do this, if you really suspect that something wrong is going on, and you really feel you need to attack the organization, then you would do an adversarial audit. But like I say, it's a pain. Usually what people do is, rather than have a direct confrontation, they will hire a third party. I've been involved in these through my consulting work. But, you know, really, the best audits are kind of a teamwork type of thing. The auditor is trying to get information about the laboratory that he is auditing, and then transmit that to the client via the audit report. Now, the client in this case can be an internal client. You know, if you're an auditor for a company, you report to your own company. Or if you're a consultant, you would, you know, there would be a client to whom to report. Now, pre-announce your intention to audit. Some people with a police mentality tell you that, well, you know, you should just show up and, you know, all this sort of stuff. But the problem is, first of all, there are some laboratories or some groups that will actually refuse to permit you entry. And you know, yeah, FDA people and other people with police authority do have the right to force entry. But if you, as a private person or a private company, try to force entry, they can kick you out on the grounds of avoiding trespass. So normally what you do is you send a letter ahead of time and kind of invite yourself over and start to set up a date. What you're looking for is a date when the audit would be mutually convenient. Now, some contract laboratories will tell you that, of course, you know, audit is never convenient. But let's face it, it's part of doing business. Now, be flexible. I mean, you know, you don't know their schedules or their workload. Don't send them a letter saying that, oh, I must show up on December 24th, you know, or else, etc. And if you want to see specific things, say so. Don't wait until you're in the middle of the audit and say, gee, I really wanted to see this. Um, you should have told them up ahead of time so that arrangements could be made. And normally you only need a day, but it depends. FDA especially will go in and have 2, 3, 4-day audits, depending upon the size of the group that they're auditing and the complexity of the questions they're asking. Now, it's perfectly legitimate to suggest dates or periods. What I will often do is in the initial letter, I will say that, I have available dates that as far would be convenient for me are the following, and I would name them and let them choose among those dates. Now, the date should be at least a month away. You don't want to get rushed into these things. Now, don't make it too far away. If you set it up 6 months in advance, there's going to be a problem because, let's face it, people forget. And but what you want to do is you want to give both yourself and the auditee time to prepare for your audit, and just work with them, because you don't want this. You want to minimize the pain of the audit. Now, the other thing is make sure you know where you're going to conduct the audit. There's a lot of people who end up dealing with they'll deal with headquarters staff. And headquarters staff can be located in a completely different city from the location that they need to audit. And what will happen is that you show up at headquarters and they'll go, the lab you want to see is located in another state that's going to take you another day to travel to, et cetera, et cetera. So make sure you talk to the head of the lab or the head of the quality unit where you're headed, and make sure, verify the location, and also make sure that you agree upon the standard that you're going to use for the audit. Are you going to do a GLP audit? Are you going to do a GMP audit? Are both of them going to be combined? ISO 17025 is getting more popular, especially on the international scale. And people sometimes do a audit based on USP requirements. And so you want to say ahead of time, because it may turn out that the lab you're thinking of auditing does not hold to the standard that you're looking at. Now, there are several regulatory standards, and in particular the GMPs. Now, there's a lot of laboratories who are sort of borderline labs that claim they're doing GLP work when actually the testing that they're doing should be GMP. And this is especially if they're doing final product release testing or anything related to actual product that's going to go out to the public. This thing needs to be GMP. And remember this, there are some new rules where FDA has relaxed some of the GMPs for early stage clinical material. But FDA has also said that, you don't use the parts of the GMPs that don't apply to early clinical material, but you will need to follow the GMPs that apply to the type of work that you're doing. And this is, of course, covered in another webinar that I give. But you need to know that any product that's going into human beings has to be tested under GMP rules of some sort or another. These rules are found in the Code of Federal Regulations under Title 21 of Code of Federal Regulations. There's drugs in 210 to 11, biologics add on 600, 610. And if you're doing devices, the rules, the quality system rules are found in 21 CFR 820. And, you know, there are GMPs for other things, cells, tissues, APIs, food, blood products, diagnostics. There are specialized GMPs that apply to these types of products. And remember, you have ISO 17025, ISO 9001. There's also WHO standards, although they're not that frequently used. But there are some international audits where you may need to invoke WHO rules. Now, remember, under the GMPs, the definition is that a QC testing lab is part of manufacturing. So anytime you see rules that apply to manufacturing, they also apply to the QC lab. And ISO 17025 is, well, it's basically 9001 applied to laboratories. ISO 9003 used to be the standard for laboratories, but 17025 seems to have superseded it. I will note that right now, most of the FDA laboratories are 17025 compliant. And there is a movement within FDA that will eventually, well, we think, will cause everybody to go into 17025 for all types of laboratory work. There are other standards, CLIA, and then business codes. Now, CLIA is the Clinical Laboratory Improvement Act, which applies to laboratories in hospitals. And they also apply to laboratories that test human samples for diagnostic work. Now, there are some of the labs who are trying desperately to avoid GMPs for reasons I don't quite understand, but nonetheless, they try to get away from it. And they will tell you that, oh, we don't do anything related to diagnostics. And this is, you know, people doing work in a clinical trial. Now, if you're not trying to diagnose the patient's condition, why the heck are you doing the testing? You know, so you would think that CLIA or one of these would be important. Now, also in some states, and these are mostly the large states, California, New York, etc., Illinois, there are state business codes that cover diagnostic testing. In fact, like for instance, the California Business Code refers to any testing on human material. It does not say it has to be for diagnostic use. It's any testing for human material. And these codes do a few things, like for instance, require that you have trained and certified lab technicians, which is kind of makes life kind of hard for people who are just using regular QC people. But you do need to look into this because you definitely want to know if that lab that you're auditing is going to run into trouble with their local board of pharmacy or health department. Most companies that are on the up and up about this will have their state licenses and certifications sort of on a bulletin board right when you enter the company. So you'll be able to see what states they're licensed in. Now, your audit team, you want technically qualified people on the audit team. And this can be sometimes, it'll be a problem, especially when dealing with labs, because first of all, you need somebody who knows, you know, auditing principles. And a lot of times that's the lead auditor. He's usually a company auditor or, well, what should become a company auditor. And this person will know about general auditing, principles and procedures, but you need a technically qualified person. You can find people with training as both auditors and in the laboratory. In my case, I was, a lab worker first and then got training as an auditor. This happens to a lot of us who eventually, you'll see in terms of, getting dragged into doing auditing. But the thing is, you need these technically qualified people because, especially with labs, it's really easy to get fooled into thinking that you know what's going on if you don't have the proper training or proper background. Now, the other thing to remember is that if your team gets larger than about 4 or 5, it gets unwieldy in terms of coordinating your activities and knowing who's doing what and when and all this. So, try to keep your team no larger than 5. Now, some companies try to save money and they say send only one auditor. And, let's face it, sometimes one auditor is all you really need. But it can be counterproductive. And the reason is that if the operation you're visiting is too large or too extensive, and you have multiple questions that you need to raise and multiple areas that you need to look into, sending only one auditor may be ridiculous because that one person cannot cover all those areas. Now, the other thing you need to do is to train new auditors. And the way you do this, and at least at the end of the training, one of the things you will do is to send this auditor out with an experienced auditor, and so that they can, literally learn the ropes. And so you may need to send a second auditor, not so much for the information or for actually doing the audit, but you need to train a second person. And this is, a fairly frequent procedure for training new auditors. And, consider that because it's very hard to just rip somebody out of a lab, for instance, and say, hey, we need you on the audit team. Go talk to Dave. He'll set you up for the travel. I mean, it gets to be very disconcerting. What you want is somebody who's participated in an audit before and, where they're sort of growing into the job. And remember, you have specialized areas, especially with laboratories. If you have questions about sterilization procedures and sterility testing and all this, you really need a microbiologist. And the other thing is, for instance, if the lab has a mass spectrometer that is doing mass spectral work for you, you're really going to need a specialist here because, otherwise it's a matter of just looking at the instrument and saying, oh, how nice, and then walking away. It doesn't really give you a good audit of that particular work. This is true with a lot of things. I mean, for instance, there are X-ray diffractometers that are used, X-ray fluorescence machines, all of these electron micrographs, that are done, and you need to have a specialist who can look at the machine and talk to the operators and know whether they know what's going on. Now, if necessary, hire a third party, go to the outside, find a contract auditor or somebody, and bring them in to cover specialized areas. 2 auditors for cost factors, I mean, it will cost more because, let's face it, 2 auditors, there's 2 separate airplane tickets and, hotel rooms and all this. Now, this will increase your costs, but speed may be a consideration. The other one I mentioned earlier is that coverage could be a consideration. You need 2 auditors to really cover a whole operation. And also, two auditors for one day can produce a report more quickly than one auditor can. And, you know, really, if you don't hire your third-party specialist to look into a special area, you may end up with a worthless audit because the important questions never got asked, or you never looked at a really important thing. And then you have a worthless audit. And let's face it, you know, a worthless product is expensive at any price. Any price you pay for it is too much. Now, there have been instances where the laboratory being audited will threaten to send a bill for the personnel time or anything involved in the audit. And let me tell you, most of the time this is a bluff. Very few laboratories actually do this, and you know, most labs build in the cost of these audits into their fee structure, so that you know the amount of personnel time that will be lost is included actually in the fees they're charging you. This used to be more common in the business. Right now, it is very rarely seen because most labs have adjusted to reality. When you get there, have an opening meeting. Now, let me tell you something about these audits. I like to show up at 9 a.m. I don't like to show up at 8 a.m. And the reason is that back when I was a laboratory supervisor, lab manager, I used to really hate audits that started at 8 a.m. because that was around the same time my guys got to work. And it made it very difficult for me and my workers to do our morning briefings and things like that. It got to be a real pain. So I always allow an hour for them to, you know, get to work, get started on their work for the day, and then I show up. Now, after you show up, have an opening meeting. Always ask for an opening meeting. You want to state the purpose of the audit. Why are you there? Are you just looking at things in general? Is there a specific area that you're interested in? You know, do you really want to see the warehouse? Is that important? If you go to the warehouse, you want to see the receiving people or how receiving samples are taken. State this during the opening meeting, because this way the laboratory people can send word down the line to say, well, you know, this guy's going to want to go out to the warehouse and he's going to want to see the area where the reagents are kept and everything like this, so that the warehouse people can have the appropriate members in place when you arrive. The lab itself should respond. It should introduce the members of its team. And a lot of places will give you a brief presentation on the history of the company or the laboratory, what kinds of testing they do, what kinds of general work do they do. And during this thing, they should give you, you know, a little bit of housekeeping details. I mean, things like, where are the restrooms? I think we've all been to parties where they feed you a lot of beer or wine, but nobody mentions where the restroom is, and you know, you end up blundering around in a partially lit area trying to find it. And you know, things like, where are the restrooms? Where are the break rooms? Where is the coffee? And then things like restricted areas. In some companies, especially biologics companies, you know, there are defined restricted areas that you cannot wander into. And even with an escort, they don't like to see outsiders in these areas. And they should tell you these. Then go over your audit schedule. And, you know, this audit schedule should be fairly loose in the sense that, okay, after this, we'd like to see this. And don't give precise times, because it may turn out that you may finish early in one area and wish to go to the next area, right away. On the other hand, it may take you longer than expected to go through an area, and you may need to take extra time. The other things, you should state is when you'd like to break for lunch. Some places break early, some places break late, like 1 o'clock lunches. And, you need to kind of work that out with your host. And, a lot of labs will provide you a light lunch. And, usually sandwiches, something like that. This is for mutual convenience, actually. It's not just to host you, but also to save their workers from having to break for lunch and then come back. So, don't expect it. This is the other thing. Every so often you will run into a laboratory or a company that will leave you on your own for lunch. And this is one of the things you really should scout out before you get there. The other thing that may happen is that they will take you to their company cafeteria and just sort of turn you loose and let you buy your own lunch there. And, one of the places this happens a lot is in hospital laboratories, where they'll take you to the hospital cafeteria, but you're going to need to pay for your own lunch. So don't go there expecting to be hosted for lunch. When you go through the audit itself, always walk through the laboratory. And this is the thing, actually go into the laboratory. Don't just stand outside and look at them through a glass window. Ask them tell them you want to follow the path of a sample. How does the sample come in? How is it received? And go all the way to the point where the test result goes out the door or goes up to whoever it is that puts the certificate of analysis together. Now, watch the techs in action, because there's an amazing number of places that will sort of conceal their techs from you. I mean, they won't let you see them in action. And if possible, have them analyze one of your samples. Now, they may not want to analyze somebody else's sample in front of you. But if it's one of your samples, there's no reason for them to refuse to do it in front of you. And again, you need to be familiar with lab operations and, look around, listen, smell, and smell. This is one of the big things. You can get a pretty good feel. Look at the lab. Is it clean? Are the techs clean? If the techs are going around with dirty or raggedy lab coats, that sort of shows you a level of disregard for cleanliness. And are there odd sounds in the lab? And does the lab smell clean? I mean, and then the other thing, remember this. Micro labs smell a certain way because of the preparation of media. And animal facilities have distinctive odors. If you've ever worked for a while in one, you know that a rat room smells different from a guinea pig room. And they all have distinctive odors. And, if they're working, well, you can expect it. If you don't smell it or it's not there, you're going to have to kind of wonder exactly what's going on. Look at the shelves. Shelves that are open on both sides really need safety lips on the shelves. This is to prevent somebody from accidentally pushing something from one side and having things fall off on the other side. And also labs that are subject to vibrations. For instance, here in California, we know that we periodically get either major or minor earthquakes. And what you want is a shelf that's set up to prevent stuff from easily it's the vibration causes the phenomenon known as walking. And you don't want your reagents to walk off the shelf during an earthquake or something. Also, are your reagents properly labeled? During a GLP inspection especially, there is a requirement that you specifically state the expiration date on the label of a prepared reagent. You should look. Do they have these expiration dates? Is it the expiration date? Is it still in date, in other words? Are the dates legitimate? Consider lab safety. Things that are shelved, you don't want them shelved above eye level. This is to minimize the chances of splashing into the eyes. And, are they properly stored? Are they shelved right? And especially things like, well, I don't have this thing here about reductants, but you don't want to have acids and bases stored together, or oxidants and strong reductants stored together, because if bottles break during an accident or something, you got a chance for a very powerful reaction that could be exothermic, in other words, an explosion. Do they have safety cabinets? Are they maintained? I've seen a lot of places that have, for instance, solvent safety cabinets. Cabinets are there, but they're not attached to any vents. And, a solvent safety cabinet with no venting is not a safety cabinet at all. So, you need to look at these things. How are the techs handling the data? Do they have lab notebooks? Do they have copies of test method SOPs at the workstation? The rule here is that you don't want to have them depending on memory to carry the data in their head from their desk to their workstation, or try to remember things in between. There should be material should be immediately available, so that it minimizes the amount of memory that they have to depend upon. Remember, even pilots go through a written checklist before they take off. They don't just mentally check off, what they need to do before they fly. And, the rule with pilots is that only a fool trusts his memory. Now, if they collect printouts, how do they handle these things? Are they stored properly? Do they take care of them properly? And, remember okay, if you look at an HPLC or GC instrument, some of these things come with a software that allows you to suppress peaks. In other words, you can cause a undesirable peak to disappear, and actually disappear from the final calculations as well. Make sure that if their HPLC has such a function, that function is disabled. You don't want them concealing peaks on you. If a laboratory is sloppy, untidy, or it's dirty, it's bad news. They don't care about their work environment. If they don't care about their work environment, what about the quality of the work that they're doing? Do they care about that? These are symptoms of increased risk of contamination, misplaced, mixed up samples. One of the things that's a real plague in the industry are mixed up samples. It's just amazing how many times there have been problems with different laboratories because of mixed up samples. Also, it points to the possibility of unnecessary variation in test results. You don't want a lot of variation in your test results. You want your results to be as precise as possible. Follow the path a sample takes. Who receives the sample? Is the documentation done? Do you get a sample number assigned right away? Is it clearly attached? Do you have a clearly defined chain of custody? Remember, in case of legal action, or for that matter, an FDA investigation, your chain of custody can become an issue. Is the sample stored properly? A sample comes into the lab. What do you do with it? You just let it sit out in the open, or do you have a place where you put the sample, and is it held there? Even this is true even for room temperature samples. They should have a location where they're kept, and that where they can be kept securely, and nobody can tamper with them once they arrive in the laboratory. Can they find the sample without having to sort through every last one that they have in the lab? There's some labs that will periodically get as many as 1,000 samples into the laboratory. And if they can't figure out where one particular sample is, it can be really bad news. Now, with biological samples, remember, these guys decompose very quickly, or worse yet, they can keep on metabolizing and destroy the analyte. One particular case that's well known is for sugar testing in blood plasma glucose. If you leave the red blood cells in there as whole blood, the blood cells will eat up the glucose. And so you have to block that. And this is normally done actually by putting fluoride into the suspending medium. So when they draw your blood, it's drawn into a fluoride containing either liquid or salt that's mixed in, just to stop it from metabolizing glucose. Is there personal interaction required for sample receipt? I mean, can anybody walk into the lab and leave a sample at any old place in the lab? There are many instances known of people coming into the lab right at about 5 o'clock or at 5:10 in the afternoon, after people have left, and just leaving the sample on a desktop or on a lab bench, and the sample sits there overnight, completely exposed and everything, with nobody watching it. Look at the sample size. Is it large enough? If you have an out-of-specification test result, will they be able to retest from the same sample? Which is permissible, as opposed to having to draw a new sample, which is not. And what about the retention samples? Is the sample large enough that you can hold extras, so that if you need to go back and reinvestigate a test result, you have enough sample to work with? Is the history noted? Incoming raw materials. When you take samples from a container, is the container marked to show that the sample was taken from that particular container? And then after the sample was taken, are the containers properly resealed? Some people take samples and then they leave the bag open, or they leave the container open, which makes, for chances of contamination. This is not a smart thing to do. So they should be resealing the samples, and this includes putting plastic seals around the necks of bottles. You want to know, when was the sample taken, when was the sample brought to the lab, and when was the sample actually tested? These 3 dates are very important. They should be in the proper sequence, and the spacing between them should not be very large. When they're running their tests, do they run positive and negative controls? At any given time when you're running a test, you need the positive control to demonstrate that the test procedure is operating correctly. And the negative control is also critical because it needs to show that you're not getting spurious results. And take a look at what they're using as the positive and negative controls. You know, sometimes the negative control especially is really a kind of a dumb control. I mean, they'll use distilled water when they should be using the sample matrix as the blank. Are they doing system suitability testing? This is the one that really shows that your system is operating properly. You know, it's not just a positive control, but is the system really working the way it should be? System suitability is defined in the USP. They have examples for HPLC, GC methods, but any test can have a system suitability built into it. Are they using Shewhart charts? Are they using statistical process control to monitor the status of the test? Now, you know, basically what you do is you take your positive control and you plot the results on a Shewhart chart. Do they have procedures for OOS results? What if the standard is out of spec? What if the control is out of spec? You know, if the system suitability value goes out of spec, do they have procedures to handle this on what happens in the event it occurs? Do they calculate final results before you discard the test solutions? You know, this is a requirement under the guidelines for OOS test result. Well, it's not a requirement. It's just that if you do this, then it allows you to go back and do your lab investigation on the original samples and original test mixtures. If you discard this stuff, then you're asking for trouble because you won't be able to recreate what really happened. You know, what other controls do you use to determine the acceptability of a test result? You know, in clinical laboratories, the positive control is run with real plasma. It's run in real plasma to mimic a true sample. The standard alone is usually not good because the standard is in a sample matrix that doesn't truly represent the sample that you're testing. And this is why you want to make sure that your controls, especially your positive and negative controls, are basically in the matrix of interest. How do they handle the test results? You know, what do? Do they handle the test results early enough so that they can check for OOS results and react to it? Who reviews or verifies? Is the review timely? For instance, there are a lot of laboratories. You get into a situation where the lab supervisor is the reviewer, except that, well, he's sort of busy and everything, so he reviews things only at the end of the day, or worse, he reviews it only at the end of the week. This is not acceptable, really, because what happens here is that you get a test result, and the test result fails, or it's unusual in some way or another, and the lab, you know, the analyst just records it in his lab notebook, and it's not caught until the end of the shift. Now, the problem here is that some of the test results need to be acted upon right away. And in this case, reviewing it at the end of the day is not acceptable. How do they review the test report? And how do they maintain data integrity? You know, if somebody makes a mistake, you know, this is what the second pair of eyes is supposed to be for. If the person transcribes incorrectly from the lab notebook onto the test report sheet, is that picked up? who picks it up and who will know? Look at the training of the analysts, at least a couple of analysts and one supervisor. You know, look at their backgrounds. The background could be weak, but if they have good in-house training, that'll make up for it. And you're looking question of seniority, level of education, and all of this. There might be a difference between supervisors and analysts in this case, but there should be a good, strong, high level of training, because a lot of this stuff, you don't do it in the lab. They have to come to you with the proper education. And no, going to the lab at some phony diploma mill doesn't really work out that well. And remember this, you can't really supervise what you don't understand. This is a thought that is kind of lost to some of the business school people. You have to be trained to do the tests that you perform. Now, nobody is trained by virtue of education or prior work. And the reason is, every lab is different, or should be different. Everybody has their own peculiarities about the way they do things. And the person needs to be trained to follow the SOP exactly. And for that reason, training has to take place in every laboratory. And, what about OOS problems? Have they had training on how to handle an OOS? Remember that analysts and supervisors have different responsibilities. And these different responsibilities are spelled out in the guidance documents. This is the old way of doing so-called training. You have the person read an SOP, watch a test procedure, and then they perform it by themselves while under the watchful eye of a senior analyst. This is considered to be inadequate nowadays. And what you want is that you need data to show that the analyst can do so as well as any other qualified analyst. And the reason for this is, if you have a laboratories where there are, say, 4 analysts who can perform a certain test, who are actually recognized as being able to perform that test, you need evidence to show that these 4 people can get the same test result. If one of these guys always gets a low result or always gets a high result, then there's a question there. It's a serious one about, their level of training and the reproducibility of the test procedure. So you need to have evidence that any analyst in that laboratory can get the same result as any other qualified analyst. Now, in some areas, you can't quantify the ability of the analyst to do certain types of work, in which case you want sign-offs by senior analysts or supervisors. And they attest to the competence of the analyst after having watched the analyst work in the laboratory. Not just the thing of, yeah, he said that he read the thing and he signed off on it, and so it's okay. That's not adequate. And FDA has not only said so, but they have cited laboratories for not having adequate training of the analysts. There should be evidence of periodic external training. Really, you get guys that sit in the same lab, and unfortunately, there's a lot of people, analysts and supervisors, who really don't want to go out for external training. They don't even like to go to meetings of groups that would force them to talk to other professionals. And the problem here is that you eventually get inbred thinking and attitudes. you start to get into this thing where people think that, well, I'm okay, you know, I've never had any problems with this test and all this, even though it turns out that they're using an extremely outdated test that has much greater variation in it than it should. And, you know, the lab needs to be aware of new developments. Now, you're going to see in FDA guidelines and everything that they tell you flat out that they expect to see a laboratory using modern methods, especially methods that are more accurate or more precise than the older procedures. So, you know, make sure that these guys are going out for training. Now, your analytical instruments should go through the standard qualification phases. You know, design qualification, installation qualification, and remember this, that some of these activities can be combined. They don't have to be in specific phases. You can combine some of the design qualification and the installation qualification, or the operational qualification with installational qualification. Some of the criteria can be mixed together. Now, the DQ seems to be a thing that drives everybody crazy. I don't know why. But basically what DQ is saying, did you know what you needed to purchase, and did you take a look at the purchase specs before you made the purchase to confirm that you were buying what you needed to buy? And it's amazing how many times this doesn't happen. And, you know, the classical one here is, well, there have been cases, unfortunately, one was in my lab, where we were purchasing a conductivity meter, and our purchasing department sent us a meter that, well, it came with a 30-foot lead on the probe. And we couldn't figure out why there was this 30-foot lead on the probe until we took a look at the meter. And we realized that the range of conductivities that it was set for was nowhere near what we needed for a lab. And it turned out, after some questioning, that the purchase that had been made was for a conductivity meter that tested the water in streams. And the reason for the 30-foot probe was that the people would stand on a bridge or something like that, or a wharf, and dip the probe into the water, as opposed to, you know, making a measurement in the lab. And this was a clear case where the design requirements did not meet up with the purchase requirements. And the IQ is, these definitions, by the way, are the ones that are found in the USP. And, you know, all of these analytical instrument qualification phases are stated out in the USP. And this is what you should be looking for. You want to know, did they go through a qualification of their analytical instruments? Now, if there's built-in firmware, as opposed, you know, firmware is the computer, the actual physical computer itself, and the input-output connections and equipment that is tied to the computer. This is the firmware. And if an instrument comes and it comes with firmware, this is the thing, you don't need to validate it like you do with software. The firmware is considered to be part of the instrument or equipment, and you qualify it at the same time that you qualify the rest of the instrument. And, you know, any changes, you go through the regular equipment change control. And, you know, remember that you need to look at performance under actual conditions of use. And I don't care how well the instrument performed in the manufacturer's laboratory. The critical part is whether it's performing well in the auditee's laboratory. That's the question you need to ask. Software should be Part 11 compliant, and they will tell you that the validation occurs during development by the vendor. I will point out to you that several FDA people have said that there is no way that a vendor can sell you validated material, because the validation, requires validation in the user laboratory or the user function. And so there's no way they could have validated it because the thing wasn't in the particular laboratory. So the people in the laboratory should have done something to demonstrate that the software meets user requirements. And at the site of use, this means the exact computer in which it's used. And they should do this as part of the IQ. Also, OTS software, off-the-shelf software. This is stuff like Microsoft Office, Microsoft Word, and the various, Adobe Acrobat and all of these things. Off-the-shelf software is software that is so widely distributed that FDA says that any problems with it, any bugs and glitches, will be well known. However, if you so you don't need to validate OTS software. However, if you modify the OTS software, for instance, with Excel, for instance, you can write little mini programs and so-called using these macros or things like that. These modifications are not validated, and you have to validate those modifications. Now, when do you need to requalify the instruments? It should be requalified after major repairs or modifications, after movement to a new location. Now, this can be within a laboratory. Well, for instance, you can have some instruments that operate near a window that is open to the well, not open, but clear to the outside. And this instrument will warm up in the afternoon because of the sunlight shining on it. And then you take the same instrument and move it to another part of the laboratory where it's right next to or right under the air conditioning duct. What you're going to find is that the instrument actually operates at two different temperatures. And the question is that do you get the same performance? So even if you move within a laboratory, you need to revalidate or requalify the instrument, usually through an OQ. Now, if you change the software, again, that needs to be revalidated. You reprogram or put a new program into your instrument. What's the performance like? Does it still do the same thing? And you have to base this on a risk assessment. And the risk is that after the event here, that you are getting the wrong answer. ICH guidance, this is on the reference standard that you're using. And this is basically the statement that comes out of the ICH reference standard. Quality appropriate to its use, it's characterized and evaluated by additional procedures. You see, this is one of the things about the reference standard. Don't just qualify or, do your measurements and everything using the method that it is supposed to be the reference standard for. You want a separate analytical procedure to confirm that the reference standard is performing properly or has the right properties. Here's how to characterize it. Here's your protocol. You write a protocol on how you're going to characterize your reference standard. Get approvals that everybody agrees that you've got the right type of protocol. You can set this up in an SOP. And, how do you execute your protocol? And remember this, utilize all the best. In other words, use the best methods available. Don't go and use a old primitive method if there is a good method available, modern method. Your data analysis and summary, make sure that's there, and that you've characterized your reference standard as far out as necessary. Now, the point I'm trying to make here is this. If you are calculating your test results to the nearest two significant figures, your reference standard should be good out to the third significant figure. Or if you're calculating to one decimal place, for instance, the reference standard should be good to 2 decimal places. And you need to make sure of this when you're looking at somebody else's reference standard protocols. What levels are they using? Now, with regard to the process or the analytical method, there's a new guidance document on process validation. And one of the things that we have been looking at is that a process validation applies not just to manufacturing, but to testing. The test method itself is a process. You have inputs, you have outputs, and all this sort of stuff. And so the process validation guidance actually has an impact upon test method validation. And what we need to look at here's a statement. This comes from the guidance document, most recent copy or version of the guidance document on process validation. And one of the things it tells you is that during your early stages, you don't need validated analytical methods at the product process development level, or when using characterization studies. However, this is one of these typical FDA documents because it's sort of self-contradictory. It just tells you you don't need validated procedures. However, it says, nevertheless, the method should be scientifically sound. And, these specific, sensitive, and accurate provide results that are reliable. This is what you do in a validation study. So first they tell you don't really need to validate it, but then they're telling you you should have data on the procedure that is essentially the same as what you would provide in a validation. You should have your method and equipment maintenance, your documentation practices, all of this, calibration practices, all of these things need to be there. And however, once you get into commercial batch release, you need full GMPs. And 210 and parts 210 and 211 are the GMPs that require that you have validated analytical methods. And, clinical supply production. Now, remember, GMP is appropriate for the particular phase of clinical studies. There are special GMPs for phase 1 clinical material. However, I will tell you right up ahead, they're not all that much different from the regular GMPs. The test methods, you look at them as a process in themselves. You should have, design considerations and what's your critical quality attribute, the CQA, for a test method. Sometimes it's not, we get test results, okay, but that's not the quality attribute. The attribute is how accurate is that test result, how precise is that test result. All of these things are defined in the validation study. And then you should be doing ongoing verification of the test methods. This is the requirement in process validation right now. You don't just validate and walk away. You have to have continuous verification. And in the case of a test method, the continuous verification can be done through SPC methods. Use control charts and look at the data from positive and sometimes the negative controls are useful here to show that your method continues to operate properly. 165E here, this is the GMP requirement that you will have accuracy, sensitivity, specificity, and reproducibility. Now, notice this sentence. The second sentence says, such validation and documentation may be, that tells you that this statement up here, this first sentence, is the validation requirement. And, this is the same as what we talked about earlier from the process validation guidance. This is 194. 194 has a bunch about the documentation, but the critical thing here is this parenthetical statement that says that if you have a method from the USP, the NF, the AOAC Book of Methods, or other recognized standard references. Now, recognized standard references are pharmacopoeias. It is not things like journals. I don't care if it was published in a journal of biological chemistry or analytical chemistry or analytical biochemistry. Those are not standard references. Now, you get into this second part of this statement, or is detailed in an approved new drug application. Now, of course, if you've got it detailed in an NDA, you do know that this test method is going to be passed on to FDA laboratories, and they will attempt to duplicate your test method and apply it to samples that come from your company. So, it's kind of crazy in the sense that if you want to send this thing out and you're not going to bother to validate it, but you include it in your NDA, there's a problem here. Now, notice if you contract with a contract testing laboratory and you're using their test method to characterize your final product, you are going to have to provide that test method to FDA, and FDA will cross-check the test method. Now here's the final statement. Suitability of all testing methods shall be verified under actual conditions of use. So this is the thing. Even with this conventional test, there still has to be a verification of suitability for use. It's not a full validation, but some kind of work has to be done and has to verify the precision and accuracy of the method. So your contract laboratory should have data, and it may not be data on your particular product, but it should be data on a closely related product with a closely related sample matrix. ISO 17025 goes through validation of methods. The validation in the ISO, may include sampling, handling, and transportation of the test material. And, if you make changes in the validated non-standard method, you need to evaluate the effects of such changes. And if necessary, conduct a new validation. You need to look at this because if the contracting laboratory tells you that it's operating under ISO 17025, then there should be data showing that these validations were conducted under these requirements. The procedure used for validation, the results obtained, and the statement as to whether or not the method is fit for the intended use. Now, intended use is testing your samples, not for testing somebody else's samples. So somebody at the lab should make a statement that, yeah, this method is suitable for use with your type of samples. Now, we're going through, these are definitions that come out of ICH Q2, which is the ICH guidance document on method validation. And these are the definitions. Notice that precision is really not defined. Precision is broken down into three components during a test method validation. Repeatability is one of them. This is same operating conditions over a short period of time. In other words, intra-assay precision. This is the easiest one to run, and it's done. It should tell you what the baseline variation is in your test method. All you do here is you set up your test method and you run a control sample. And I do mean control sample, not standard. Standard proves nothing. It has to be a sample that resembles the real sample. You run this, like, say, 6 times, and you calculate the standard deviation of the test results. And this gives you your repeatability or intra-assay precision. This should be the minimum variation that you see in your system. All other variations should actually be higher than this because they will incorporate this variation. Intermediate precision, these are laboratory variations that actually have nothing to do with the test itself. But these are variations such as the day-to-day variation, analyst-to-analyst variation, different equipment. You know, if you have 5 different HPLCs that can perform an assay, do you get the same test result? What variation do you see among the 5 different instruments? You know, there's one thing here, different analysts, and this is one that bugs me all the time. You tell people that you need to see different analysts, and what they do is they take only 2 people from the laboratory, usually the senior analyst and somebody who's almost as senior as the senior analyst, and they'll say, well, okay, you know, Mary and Beth will run the 2 tests, and we'll compare their variations. That doesn't prove anything because Sam may be the actual guy that runs the test for you. So you want to see what happens when Sam runs the test. And so you want to compare variations among all of the qualified analysts in the lab. You know, the best way to study this is you use what are called design of experiment methods. Fractional factorials, and, well, you know, simple ANOVA methods, analysis of variance methods, will allow you to look at the different analysts and see if everybody's doing the same job or if there's a particular analyst that has a problem. Reproducibility, it's the precision between laboratories, and this really is a European definition. This wasn't the old American definition, but basically it's lab-to-lab variation, and this is done through collaborative studies. Again, you can use ANOVA methods to study this. If you have a company with more than one laboratory performing the same test, you need to compare your test results among different laboratories. And this is usually done by passing a set of standards or controls among the laboratories and then looking at their test results. Accuracy measured by comparison, response to which a blank sample to which a known amount of analyte has been added. You know, question is, do you get what you expect to get? And You can also do this by using so-called orthogonal methods. You have a test procedure, for instance, that measures protein. And you have a different test procedure that also measures protein. You use one as the reference procedure and one as the test method, which is probably the assay that you're looking at. And you ask the question, do you get the same test result from both methods? Or does your new method give you as good as a test result as the old method? And this needs to be looked at. Now, by orthogonal method, I mean things that are completely different in their chemical procedures. For instance, if you're using a UV method, okay, for measuring protein, you do realize, of course, that the UV method that is being used normally depends upon the amount of aromatic amino acid that's present. There are tests, such as the Lowry test, that are also protein measuring procedures that also depend upon the amount of aromatic amino acids present. So these are not orthogonal. However, a Biuret method that depends only upon the presence of peptide bonds, that one would be an orthogonal method. So you need to look at this. You need to make sure you're using orthogonal methods when you make this comparison. You can do t-tests, the statistical t-tests that compare two test measurements to see if they're really the same and all of this. Now remember, accuracy, precision, all of these things have to be measured at concentrations high, middle, and low of expected values. Now notice this, I say expected values. So normally for an analyte, you will have an expected range. And the whole thing is that there should have been a test on the high side, there should have been a test on the low side, and also one in the middle. This is to verify that the test works the same way, regardless of the level of the analyte. You know, testing at a single concentration doesn't prove all that much. Specificity, the thing about specificity is that you have to, now notice the thing, presence of components which may be expected to be present. These things, they're things like process impurities, residual solvents, denatured, degraded active ingredient, or excipients. You know, these are things that come out of the process. These are things that are expected to contaminate your sample. Now, one of the things here is that, you know, your denatured, degraded active ingredient or excipients. And also remember, it is possible to get a product where the active ingredient has reacted with an excipient, or excipients react with each other, etc., etc. These are degradation products. And the place you find these is in the stress testing portion of your stability study. This is one of the things. See, you need to do your stability study so that you know what kind of degradants can be formed in your samples. And you take these and verify that they don't affect the outcome of your test. You know, you need to know this. And at the same time, you need to know, for instance, in some protein assays, for instance, a partially degraded protein still shows up as protein in the assay, despite the fact that the biological activity is completely lost. So if you're trying to couple biological activity with protein concentration, things can go very wrong because of a lack of specificity. Limit of detection, LOD, Always remember there's an upper limit of detection as well as a lower limit of detection. And there's some rules here about multiples of noise and all this. These rules are found in the USP. They're not considered by analysts in a way to be very good methods, because these are just sort of rules of thumb. And what you really need to do is you can set it up initially when you're first developing your method. You can set up your limits based on this, but you really need to verify these things by direct measurement, and you can do this. Now, if you don't have detectable noise, well, for instance, if you're running an HPLC study and the concentration of your analyte is so high that you don't have to operate the instrument at maximum sensitivity, so that the sensitivity you operate at essentially gives you a flat line, a flat baseline with no noise in it, then you have trouble establishing a limit of detection that's based on noise. And what you do is you look at the confidence intervals of your test results and see where it no longer includes 0. Your limit of quantitation, usually you get this from a direct plot. Again, you can do it by using the USP method, which talks about noise. But again, if your method has no noise, then you get it from a straight line plot. And there's an upper and lower limit of quantitation as well. Now, these things should cover the same range as what you used in the repeatability study and the accuracy study for that matter. Highest and lowest values. Now, remember this, if your analyte concentrations are not limiting and where it's not of concern, LOD and LOQ may have no meaning. Usually LOQ on the low side, anyway, is important when you're trying to detect impurities or toxic material, that sort of thing. But in some cases, you really don't care. And so you wouldn't need to include it as part of your validation study. Linearity is basically over a given range to obtain test results that are linear, directly proportional. Now, your linearity is defined over a given range, which should be the same, range that you just talked about in the earlier slide in terms of limits of quantitation. Remember that, well, the definition of linearity is given by a term known as a correlation coefficient, which in standard statistical nomenclature is R. It is not R squared. R squared is known as the coefficient of determination, and it's slightly different from what R is. And there's a bunch of really, cell biologists getting into analytics and things like that. There's people out there who look at the definitions in Excel. Microsoft has really followed up some of the definitions in Excel. And by looking at Excel, they think that R squared is the definition of linearity. And they use R squared. So, kind of take a look at this, because it tells you, it gives you an indication of the level of statistical sophistication that the laboratory is using. Remember this, a line doesn't need to be straight as long as it's predictive. There are curves where you've got a good equation for the curve, and that curve becomes predictive. And the other thing you can do is you can do transformations. Linear values or so-called linear values can be converted to logs, makes them more linear. Log, you plot the logarithm of concentration versus the logarithm of response. inverse 1 over concentrations, 1 over the response, the square root of the response. There's actually a thing called the arc sign that has also been used for linearizing results. The thing to remember here, if these transformations are used, the linearity exists only in log space. It does not exist in normal linear space. And so when you're looking at the test results, it's fine. Everything, log and all that. But when you try to translate these things back into normal space, you can end up with some strange numbers. So stick with, if you're operating in log space, stay in log space. The range. The range actually is the distance, is the straight line portion of your linearity study. And, it's that interval. Now remember this thing, when you, high, middle, and low, the range corresponds to high, middle, and low in your studies for your concentrations. The high value should correspond to the high value of the range. The low value should correspond to the low value of the range. And the middle point, the midpoint, halfway between high and low, should be your expected concentration or your midpoint. You can use these to justify each other. And remember, your limits of quantitation, your linearity, and your range all should fit together. And you can use these, or they should be using them. Robustness is now, these are actually test parameters. For instance, you say the sample must be at a certain pH. And most people, sensible people, will say that, okay, the sample has to be at pH 7 plus or minus 2, or plus or minus 0.2 rather. Okay, so in your robustness study, you want to set up a thing where you run this at pH 6.8 and also at pH 7.2 to see. I mean, this is your allowable range. Now, prove to me that samples at those extremes will behave normally in the test. If you say that the sample should be in normal saline, what's your spec around that concentration? Some people go high and low on their saline concentrations. What's your allowable range? If you say that the water bath should be at 37 degrees, is it plus or minus 1? Can you control to plus or minus 1? In which case, can you tell, is there a difference between 36 degrees and 38 degrees? All of these things, these are assay parameters, and you're allowed to have some variation in them because, let's face it, nobody's perfect, and so you will get some variation. Sometimes, like with your incubators, it's going to be because of the instruments themselves. 37-degree water bath turns on at 36 degrees and turns off at 38 degrees. And it kind of tries to coast at 37. So there are these ranges, and whether you know it or not, they're there. So you need to deal with this, and this is what is done in your robustness study. And there's a whole bunch of these things. Remember your volumes pipetted. Excuse me. Your reagent concentrations. You say you have a reagent at 0.1 normal, but is it really? Is it like 0.1 plus or minus 0.05 normal? Oh, excuse me. I seem to be getting attacked by an allergen of some sort. The best way to study this is, again, through design of experiment methods, particularly things like Placket Berman or Taguchi designs that allow you to eliminate things. Now, there are people around who will do a lot of what is called, experiment by waving of hands. And these people are going to tell you that certain things are not important. Well, you need proof that these things are not important. And that's what you do with some of these designed experiments. Now, here's the thing. For an identity test, all you need to validate is the specificity. That's the only element that's required. A limit test for an impurity, you only need specificity and limit of detection. It's the quantitative test for an impurity where you need everything. Assay for an active substance. Now notice this. In an active substance, you're not particularly concerned about the concentration. So you really don't need to do things like limit of detection on this thing, because you should have enough, let's face it, you better have enough active substance. When you're closing the audit, ask for a period where you can sort of gather your thoughts. Even if you're by yourself, ask for about 15 minutes. Organize the closing presentation. Make a decent presentation at the end. If you have a group, the lead auditor is in charge of this whole thing. And, think of at least one positive thing that you can say about the organization. Now, I will tell you, I have been on audits where we had 4 people sitting around a room for 15 minutes trying to think of something positive we could say about the company. And the only statement we could come up with was that we really appreciate the landscaping in the front of your building, which, of course, they didn't even have anything to do with. You should have your escorts, and the head of the quality department should be there. And along with supervisors of the functional areas that you visited. Now, here's the thing. Nothing in your final audit report should come as a surprise. So make sure you go over the important findings during this closing meeting. The whole purpose here is to give them a chance to informally respond to some of your findings, because it may turn out that some of your findings, and let's face it, everybody does this, you may be jumping to a conclusion, and you want them to have a chance to defend themselves and explain to you why that thing is not particularly of importance. Don't let yourself be squeezed for time here. Make sure you've got enough time. Don't get into a situation where you're going to have the meeting at 4 o'clock and at 4:30 you have to leave for the airport, because they can kill you by dragging out the meeting. Always allow yourself a lot of time, and make sure you talk about how you feel about serious items, and just don't blindside them. If you don't discuss these things, don't put them into the audit report, and then close on that positive note, that positive thing that you thought about. Use a standard format for your audit reports. I've got all the elements right here. And then send a draft copy to the auditee and ask them for corrections of factual errors. Now, the thing is, some of these guys are going to try and start a debate on you right away. Don't accept that. This is not the time for a debate. This is the time to correct any factual errors. And then finally, when the report is final, make sure you send a hard copy to the lead auditee at the lab that you audited, and then have email copies to all supervisory people. Ask for a response by a definite date. You know, FDA only allows you 15 calendar days to respond. So if the response is unsatisfactory, better say so, and have an audit file for the laboratory. You know, for every laboratory that you are contracting with, you should have a file. And that file should contain things like their advertising brochures and your audit reports. And before you go out on an audit of that laboratory, you should review what's in that file. And, you know, and make sure they've done the corrective actions that they promised and all this. If they haven't, that's an item for your audit. You need to look at that when you get there. So, finally, a little bit over, but it's time for questions, I guess.
All right. Thank you so much, Dr. Guara, for your session here. Ladies and gentlemen, like Ms. Guara just said, we'll go ahead and start off the question and answer session. So if you have any questions for our speaker today, this would be the time. What you could do is to verbally put forward a question for Dr. Guara. You could go ahead and click on the raise hand option, which is a palm-like icon on your participants panel, for us to go ahead and know that you have a question. Then I can go ahead and unmute your lines, and then you could directly post your question forward. Or you could post your questions on the Q&A panel, and then Dr. Guara and myself would be able to look through the questions, and he should be able to directly answer the question. Or if you're not able to do either of the two, you could directly send me a chat message with your question, and I'll post it forward for an answer. So if you have any questions, please do go ahead.
Also, before we get into this, or while we're getting into it, I will make the note that if in a day or two, you know, usually the next day you think of a question you should have asked, please feel free to forward that question on to Michael, and Michael will transmit the question to me, and I will answer it. I won't guarantee instant answers, but we will get back to you. So if you don't have a question now, but you have a question later on, feel free to bring it up.
All right. Thank you so much. Like Dr. Guara said, you can definitely post your questions forward if you do come up with questions after today's session. Dr. Guara, I in fact do have a couple of questions for you here.
Okay.
One of the questions which I see on my chat message here, the question is, could you elaborate on FDA direction for ISO 17025 guidance?
Okay. You have to realize this is not official. However, I have talked with FDA people, and they say that there are, well, first of all, FDA has gone out and certified their laboratories against ISO 17025. As I understand it, last year or the year before, they finally got the last of their laboratories certified. The statement I heard was this: once FDA has gotten their lab certified and they have some experience in dealing with it, they feel that they will go and require conformance to 17025 for all laboratories, not just testing laboratories, but you know, laboratories doing development work, R&D work. You see, this is one of the problems, is that they want to get back early into the R&D work and the development work and regulate those laboratories as well. And apparently 17025 provides a mechanism for that. It also covers things like testing, test methods, all of that. And they may just go and adopt 17025 as the procedure to follow. Pretty much the way that they took ISO 9001 and incorporated it into the device GMPs. they're looking at 17025 for all laboratories. Now, I was told this sort of on the side during a dinner. And so, like I said, this is just what I was told by one FDA lab worker. And it's definitely not an official position.
Thank you so much. And Dr. Gore, I do have another question for you. The question is there ever any third-party auditor legal liability?
Yes. Depending upon the material, you may end up in a chain of audits where you're, well, for instance, if you go to a contract laboratory that is using a sort of a specialized instrument, say you have say they've got an X-ray fluorometer. Question is, when they purchased that X-ray fluorometer, did they do a design control? Did they a design qualification? Did they verify the quality system that's followed by their instrument manufacturer and all that? And you should be able to expect that they have this paperwork on file. Another one, for instance, is in the case of an animal testing laboratory. If you go there and they're using a specially blended animal feed for their animals, for instance, not everybody buys stuff from Purina. There are regional feed companies that'll make custom blends for your particular laboratory. But then the question is, did they go to that manufacturer and verify that there are controls on that material that they purchased from them? And so there could be a chain of audits where you audit a lab and then look at their audits of their contractors. And this is, well, it wasn't so common in the past, but it is becoming more and more common now, where people are looking at these things.
All right. Thank you, Dr. Gore. And ladies and gentlemen, we'll give it an extra 30 seconds. If you do come up with any questions, please do go ahead. And while you do so, I just want to remind you that the feedback form is open in the polling panel right now. It's got about 8 questions, and these are multiple choice in nature, and it shouldn't take you more than a minute to answer. And we would really appreciate your feedback and value them. The reason is not just your positive comments, but even if you have any negative feedback, it would help us to improve our sessions, not just Dr. Guara, but the team at Global Compliance Panel as well. So if you do spare about a minute of your time, and if you can just go ahead and give us a feedback, we would really appreciate it. And at the same time, on screen as well, you would be able to see the past webinars, which are available in a recorded format of Dr. Kuwara. In case you are interested, you could absolutely go ahead and give us a call, or you could log on to our website, and you also see the link available as well on the page, and you could click on the link and it should direct you to the Dr. Kuwara's page on the Global Compliance Panel website. And at the same time, there is a webinar which is coming up of Dr. Kuwara, and this is slated for the coming month. So if you are interested as well, you could absolutely give us a call or log on to our website. And Dr. Kuwara, I do not see any questions being posted. So we'll go ahead and conclude the session, but before we do, any remarks, any concluding statements for our attendees today?
No, I guess thank you for being here, and I guess basically I'd like to make sure that it's clear in your mind that you can ask us questions at any time, that if you want to follow up on this presentation, feel free to contact Global Compliance Panel and ask your question.
Absolutely. Thank you so much, Dr. Kuara. And ladies and gentlemen, I just want to thank Dr. Kuara and all of you all for your participation today. And you can see our numbers on the screen and the addresses as well. If you are interested, you could always, if you're open for any kind of suggestions and feedback as well. And today we'll end the session, and I really appreciate all of you all taking part in today's session. On behalf of Dr. Kuara and the Global Compliance Panel team, I'd like to thank you all and wish you all a lovely day ahead. Thank you so much.
Okay, thank you, Michael.
Thank you, Dr. Kuara. You take care of yourself. Hope everything's fine at your end. All right.
Yeah, everything's good. Looks like the storm has cleared up a little bit, so
Right.
I will see you later.
That's a good thing. Absolutely, Dr.
Bye.
Kuara. I'll talk to you in a while. Thanks so much.
Yeah. Bye.